Ma'am, can we start or we should wait 1 or 2 minutes, please? Renuka ma'am?
Please start, Sakshi. I think start.
Okay. Indrani, are you recording?
Yes, ma'am. I'm recording, ma'am.
Okay, thank you.
Thank you, ma'am.
So, good evening, everyone. Today we are going to start for our WeCare webinar. And today we have a uh, keynote speaker, Dr. Hima Kumari. I'm going to introduce her with her very beautiful scientific career. She has obtained her master's and doctoral degree in genetics from Osmania University, Hyderabad. She has completed her Dr. D.S. Kothari postdoctoral fellowship from JNTU, Hyderabad. She worked as a research associate at A-Star Singapore for developing plant stress protein database. She has 18 publications in various peer-reviewed journals with a cumulative impact factor. She has also received a gold medal and a 4 Young Scientist Award, Wired Award for oral presentation in various national and international conferences. Dr. Hima has received Professor K. Vaidyanath Memorial Young Scientist Award in the year of 2019. She has received Best Research Paper Award from Indian Botanical Society in 2014. She is an Associate Fellow of Andhra Pradesh Academy of Science. She is a life member of Indian Science Congress Association and Bioclues, and also she is a member of Asian PGPR Society. She is well versed with plant molecular biology, biochemistry, tissue culture, and bioinformatics technique. And also is a beautiful mother of 2 children. So I would like to request to Dr. Hema that she is going to present her uh, on her topic, and her topic is genome-wide analysis of sodium ion and potassium ion transporters, which is a crucial player in a complex game of salt stress tolerance. So, Hima ma'am, please share your screen and enlighten us with your special topic. This, thank you.
Yeah. Good evening, everyone. Thank you, Sakshi, for introducing me with a beautiful note. Uh, and I would like to thank Dr. Renika for inviting me for giving the presentation on this occasion for weekend webinar. So today I'm giving a presentation on my PhD and postdoctoral topic, which is a genome-wide analysis of sodium and potassium transporters, which are very crucial players in the salt stress. Just a second. Hima Proton. Okay. So my PhD work and postdoctoral work we have done majorly on the abiotic stress factors on the salt stress. So today I'm presenting on a topic of genome-wide analysis of sodium and potassium transporters, which are a very crucial players in a complex game of salt stress. So, as plants are sessile in nature, they cannot move, so they are severely affected by the various abiotic stress factors like salt, drought, flooding, heat, and various abiotic stress factors. Together, these stress factors they impose a various series of morphological, physiological changes which are affecting the plant growth and productivity by almost 50% of every year. So, of the major abiotic stress factors, I'm presenting today on a salinity. So, plant needs a more of potassium than sodium because sodium is toxic at the higher millimolar concentrations. Why plants need potassium? Because they are very essential macronutrients and it's necessary for various cellular processes like enzymatic reactions, photosynthesis, protein synthesis, regulation of ion homeostasis, and cell elongations. So this ratio of sodium and potassium in plant cells they are maintained by various transporters belonging to the cation, proton, antiporter families. So out of these families, we are worked on a sodium and potassium transporters. Uh, my work is mainly on a sodium transporters, and my colleague worked on a potassium transporters. So these transporters they transport sodium either out of the cell or into the vacuoles through various mechanisms like selective and non-selective pathways. So one of the major pathways which are associated with the sodium and potassium uh, pathways is like when higher calcium uh, sorry so when salt stress is more the calcium sensors they activate the SOS3 and SOS2 transporters which together form a complex and they uh, activates the SOS1 which is present at the plasma membrane levels. So this SOS1 transporter it excludes sodium out of the cells and at the vacuolar level sodium is compartmentalized into the vacuoles. So these channels they maintain the ion homeostasis and vacuolar and so plasma membrane pathways are present. So what we have done is many sodium and potassium transporters are present in a plants. So we have done genome-wide analysis in a sorghum bicolors. So which reveals that out of the 7 transporters, 2 are present at the plasma membranes and 4 are present at the vacuolar. One is at endomembranes. So far, only sodium transporters, antiporters were found in various plants like Arabidopsis, Ziamis, rice, or but for the first time, we have taken the homologs from the humans also, which are exchangers, sodium exchangers. So in humans, 9 sodium proton exchangers are present. So we thought these whether these homologs are also present in a sorghum or not. So these homologs are also present when we identified and total 17 transporters were identified in sorghum. And whereas in case of potassium transporters, total 47 transporters were identified, out of which 27 are HAC, high affinity potassium transporters, and 4 HKTs, and potassium uptake channels, AKT, and 2 TPC are present. So for the first time, we have done the genome-wide identification for the sodium proton exchanges and antipoters in sorghum bipolar's, and along with the Arabidopsis. And this work was published in a Genes from the MDPP group. So what we have done is 17 transporters were taken, and this we have done a gene structure analysis. Like after this, the introns 0 to 9 introns were present in the all 17 transporters. And we have done a motif characterizations also. For these sodium proton exchanges and transporters, amiloride is a way specific functional domain is present in the sodium transporters, which is nothing but it is an inhibiting binding domain of the sodium proton activity. So this amiloride binding signature sequence, which is shown in the black color, it is present in all the sodium proton exchanges, whereas NHX 4, 6, and 7 and NHX E, these four genes, they doesn't contain the amiloride binding site. We have also done protein modeling of the sodium transporters for all the 17 transporters using a ITASR model, and we have obtained that this amiloride binding site and other sodium proton activity domains are also present. And we have performed a protein-protein interaction analysis for these sodium proton exchanges and transporters, and what we have found is NHX5, it is found to be interact with all the transporters, maximum of the transporters, and is involved in the salt stress pathways like NHXE, NHE5, and other transporters in a sorghum bipolar. And when we have performed the phylogenetic analysis of these transporters among the sorghum bipolar, Arabidopsis, eucalyptus, and other plants, what we have found is these transporters are present at the three main localizations like plasma membrane, vacuolar, and endo. And these are all falling under the three cluster groups. And you can see this sorghum bipolar NHX1, it is the ortholog of the Ziamase, and these are present in the vacuolar, and it's compartmentalizing the sodium into the vacuoles. And we have also found that through physical gene mapping for these transporters, we have found that three gene duplication events. You can see at SBNHE1, which is showing with a green color. Through duplication events, it is found in the other chromosome, like it is present in the chromosome 8 and it's also in the chromosome 1, but with the different functions. So duplication events were found, and these genes were found to be paralogous, and they are diverging in the same species. And with the homologous gene mapping for sorghum, rice, and Ziamase, we have found that the NHX2, 4, 5, and 6 in Ziamase, sorghum, and rice, they are present in the same chromosome. You can see here in sorghum it is present on the chromosome 2, and in rice it is on 7, and in maize also it is on 7. So these are the genes which are present on the same chromosome, and they are found to be the hotspot regions for the salt stress signaling pathways. And we have also to check the functional divergence of these genes NHX and NHE exchanges under the different stress conditions. What we have taken is we have taken a sorghum bipolar tissue, like different tissues, embryo, root, stem, and leaf of, and treated with the different stress conditions like salt, drought, heat, and cold under different temperature. Like salt, we are treated with a 200 millimolar, drought with 200 millimolar mannitol, heat at 42 degrees, cold at 4 degrees, stress conditions. And upon the different treatments and with the different tissue conditions for these all transporters, we have performed the real-time analysis. We have taken, after treatment, we have taken like isolated RNA and converted into the cDNA, and with the different genes we have performed the real-time analysis, and you can see here the different variations of the expression levels. Uh, this light green color is showing it is like under drought. Embryo tissue is showing a low specific expression, whereas this all red color dark conditions, this SB NHX7, exchanger 1, 2, 3, and 4 under salt stress of the stem and drought, like stem under different treatment conditions like salt, drought, heat, and cold, it is showing a more expression levels, whereas embryos and you can see here leaf, these are all showing a low expression levels, whereas in stem it is showing a low expression levels under native conditions. Apart from the sodium transporters, we have also performed the genome-wide analysis for the potassium transporter gene homologs. Similarly, the exons are presented in the red boxes and introns in a black lines. So we have taken 47 transporters from the sorghum bicolor. So different groups of families for HKT, KEA, potassium channels, and HAC family, we have found that conserved motifs of these families like proteins. You can see the different consensus sequences from like 20 up to 20 amino acid sequences were performed. And we have also identified the number of cis elements like in different classes of promoter elements for each, you can see, and we have categorized into the three different groups like abiotic stress responsive elements, biotic, and phytohormone groups. So under these abiotic stress relative elements, almost 1759 more number of cis elements were found in the MIP transcription factor family, whereas low number like here 28 is found in a CRT and triple one is found in the ARR1. So these are all transporting cis acting elements were found in the potassium transporter groups. So we also performed the protein modeling for all these potassium transporters 47, and we have identified the conserved domain sequences. And this is a string protein interaction family. You can see SB-HAC1, it is interacting with a more number of proteins. And we have found the phylogeny tree of the potassium transporters for sorghum, rice, and Arabidopsis, and it is showing the different orthologous genes. And through physical mapping activity of these potassium transporters, we have found the 8 duplication events for this. HAC1 with the AK2 in chromosome 2, and whereas HAC7 with the KEA1 in chromosome 6, HAC6 again with the HAC13. Like 8 duplication events were identified in sorghum itself. And we have also done the synteny analysis of these potassium transporter genes using a Sarcos TBS elements. You can see different groups were falling in this. Similar with the sodium transporters, we have also performed the different treatment conditions for root, stem, embryo, and leaves using the various conditions like a drought, salt, heat, and cold treatments. And various expression levels were identified for the itself. You can see in a hack it is showing a more expression levels for a stem and leaves. And my PhD work was actually on the like sodium transporter. It is a sodium transporter like protein which was a plasma membrane bound. We have isolated from the sorghum bicolor and expressed in a tomato for maintaining the ion homeostasis, and this was published in a Frontiers in Plant Science in the year 2017. So this is actually my PhD work which I have done in almost 5 years back. So what we have done is SB-NHX protein which is isolated from the sorghum bicolor, which is a drought tolerant variety. It was taken. It is the size of this gene is 1473 base pairs, and it is cloned into the picamidea vector, which is, and you can see the construct of 2.3 kb. and this is the amyloid binding site it was found, and this is the PKMBF 1302 vector we have identified, and you can see it's with the hygromycin selection marker, and it is also have a GFK as a localization. This is the gene which are constructed, and we can digest with the KPN1 and BAMH1. And after that, we have standardized the tomato PED variety we have taken, and we have taken different stages of regenerations. So this gene was cloned and expressed in a different explants and standardized. We have taken cotyledon and hypocotyles, and this is a multiple shoot induction. So and rooting, they are transferred into the polyhouse. Finally, they are growing in a net house. So after standardizing that, we have characterized the transgenics. So we have isolated the genomic DNA isolation with the NHX-LP. We have confirmed this is a partial gene size 776, and we have also confirmed with the hygromycin selection marker. This is a southern blotting. So you can see this is a copy number. Two copy numbers, single copy numbers. So for further physiological and biochemical analysis, we have taken the single gene copy number plants. So what we have done is the single gene copy number plants, four transgenics were taken, and they are treated with the 200 millimolar sodium chloride treatment for 72 hours. After 72 hours, we can see that the control plant showing a yellowing and slowly it is wilted, whereas the transgenic plants are showing a better performance and they are well tolerated even after the 72 hours. So since this gene is a membrane protein and it's localized at the plasma membrane, to confirm with that, we have performed the immunolocalization experiments. So we have taken the control and transgenic plants, uh, these transgenic plants, and conjugated with the elixir antibodies, and you can see this control is not stand, and the transgenics you can see there's a its gene is located at the plasma membranes. And after that, we have also performed the calcium green indicators because this gene is excluding sodium out of the cell. So to check that after excluding sodium out of the cell, the calcium comes into the cell. So what we have done is we have also stained with the calcium green indicators. You can see here. So the gene is showing the calcium green in the plasma membrane levels only. So this indicates that the sodium is excluded out of the cell after the salt stress treatment. So we have also done the like root and stem anatomical studies for the one transgenic, and you can see here the untransformed control and the transgenics. The transgenics are showing the thick wide lumens and thick vessels, whereas the roots are showing like the compact cells. Whereas the wide lumens are found in the transgenics roots and stems, and its solitary vessels are found, which clearly indicates that they are in like included in the xylogenesis, which is means that it's a in which performs the water conductivity of the cells even in the salt stress conditions. And we have done a ion analysis of the sodium, potassium, calcium, and chloride ions in the control and transgenic glands. So after treatment with the 200 millimolar for 72 hours, we have taken a different tissues like root, stem, leaf, and flower. You can see in a wild type the sodium is more, whereas in the transgenics the sodium content is decreasing in a leaf and in flower also. Flower doesn't show much difference between the wild type and transgenics, but the stem roots, like you can see, they are showing a very less sodium, and compared with the potassium, this potassium amount was increased in a transgenics compared to the wild type. There was no much difference was observed in a calcium in a wild type and transgenic plants. So we have also performed the biochemical analysis like for antioxidants like proline, SOD, catalase, and we have also performed the chlorophyll fluorescence. Proline was almost fourfolds was increased, for 15 days we have performed for like at 0 days. First day no much difference was observed. At fourth day the proline content was increased by 4 days, and at the seventh day also, but gradually after 15 days the level was decreased. Whereas the SOD activity was, there was no much difference was observed, like only 1.5 fold was found, and catalase activity it was showing a 2.8 folds was increased in the transgenics compared to the wild type in different tissues. Whereas chlorophyll fluorescence analysis we have treated after treatment for 72 hours, we have checked the chlorophyll analysis fluorescence activity of PS2, like using a handy P instrument at the Hyderabad Central University. So we have found that after treatment, wild type was decreased, the activity was decreased by 48%, whereas in transgenics only 15% was decreased. So which indicates that these plants, transgenic plants, they have shown the better tolerance activity. So since this proton family, like cation proton antiproton family, it has almost 3,800 transporters. So what we thought is whether this protein is really interacting with any of the proteins or not. With the bioinformatics studies, we have found that NHX5 and other proteins, they are interacting with many proteins. But we have performed for the first time, like we have validated with the wet lab experiments, and we have performed the protein-protein interaction using the co-immunopresentations. What you have done, you can see SB-NHX-LP. This is interacting with the many of the proteins. So, using a co-immunopresentation, we have found that it is interacting with the CHX2, cation proton antiproton 2. So this protein was identified uh, using a SBNHX antibody, and we have done like a mascot analysis. This is the page we have performed after that. So you can see the wild type is not the transgenics, it is showing SBNHXL, and it is uh, CHX2 protein. It's a 75 kD almost its size. So after angel indigestion, we have performed this MSMS analysis, which is interacting with the showing the protein CHX2. So this indicates that SB-NHX-LP is interacting with the CHX2, which is involved. It is mainly involved in the potassium acquisition into the plants. So QRT, like RT-PCR analysis, also it indicates that it's SBNHX-LP and its interactant, whether these two proteins, after immunopresentation, these two proteins, whether they really showing any variation under salt stress, salt, drought, and potassium nitrate and potassium chloride. Different treatments were given to the root, stem, leaf. So SBNHXLP, it is showing a 4.5 folds and 2.3 folds in root and stem respectively, whereas CHX2 it is also more like almost 4.7 and 4 folds was increased under treatment of sodium chloride and potassium nitrates, whereas drought it only shows 1.7 folds for a CHX2 protein. Contrarily, it is showing that potassium chloride treatment it is only slightly inducing this activity of NHX-LP, whereas CHX2 it is showing almost three folds increase under sodium chloride. So since we know that this like amyloride is inhibiting the activity of NHX, and we have performed that also in biochemical experiments. So we want to know whether apart from the amyloride binding sites, any other inhibiting sites are present in NHXLP or not. So apart from amyloride, we have found other four proteins like drugs, benzamyl, carbonate cyanide, and MIA and TPA. They are also found to inhibiting the activity of NHXLP, but we have not done the like we have not proved through wet lab experiments till now. We have confirmed with only docking studies. So coming to conclusion, like sorghum bipolar, it contains NHX 7 transporters and 9 exchangers, whereas it also has 47 potassium transporters, which are uh, expressed mostly under the cold, salt, and high temperature stresses. During my PhD work, I have isolated the like 1.5 kb of the NHXLP from sorghum, which is nothing but a plasma membrane bound protein, and it is confirmed with the co-immunolocalization, immunoprecipitation experiments, and different studies. For the first time, this gene was isolated and it is showing the sodium exclusion at the membrane level, and it is also helping the transgenic to ameliorate the salt stress by interacting with the cation proton antipotent 2. So, finally, I would like to acknowledge various funding agencies like DST during my work, which is a part of the project work in the DST for 3 years. After that, I continued for the PhD work, and after that, I have received the fellowship from UGC in the form of JRF and SRFs. And I worked as a CSR RA for this genome-wide analysis, and I continued this work for my postdoctoral studies, like to know the activity of the CHX2. So we have carried out this genome-wide analysis in the ICRISAT. I would like to thank Dr. Tushar Shah, Santosh Deshpande, and Dr. Rajiv Varshne. Uh, and also we have developed the protein stress protein database in collaboration with the A-Star at Singapore. Uh, we have received the travel fellowship in the year 2012, which I would like to greatly acknowledge Bioclues, Dr. Prashant, for helping us in carrying out these research experiments and various bioinformatics studies. And finally, I would like to thank and acknowledge my PhD guide, Dr. Professor Kavkishore, who has given all this guidance, and he helped us in a thin and thick various supporting, and he used to give like various uh, ideas to how to carry out this research work and how to improvise that one, like that. And I would like to thank my colleague, my friend, and my better half, Dr. Anil Kumar, who is always there with me, and he supported me for my PhD and PDF works, everything. Thank you.
Thank you, ma'am. It was a very wonderful presentation, and you have means like in your work, I have seen that you have in your work genomics and proteomics studies both are involved. So it's very great. I would like to request to every attendees to ask a question to Hima, ma'am, please. Please, you can ask questions to Hima, ma'am.
Hello, ma'am. Yes, ma'am. Hello, ma'am. Is my voice audible?
I would again request to all the attendees to ask questions to Hima, ma'am, please.
Yeah, Hima, actually, it's an excellent talk and a lot of things involved with respect to sodium and potassium, like stress with respect to plants to have vital proton. When coming to docking studies, and have you tried to see? Are you able to hear me?
Your voice is a bit low, Renu.
Yeah. Yeah. Is it okay now?
Yeah.
Yes, perfectly fine.
Okay. Yeah. So with respect to docking studies, yes, done so like have you tried to look at the binding energy and to kind of stability studies with respect to the sites where you have been identified that there is a binding with the residues?
Renu, sorry, could you please repeat the question because its voice disturbance was there.
Can you hear me? Yeah, I was just going into the docking studies where you were able to find there some residues where they are actually binding. But
Yeah.
how about binding energies and their stability? Are you able to look into those things, or how it is?
Uh, we have actually the amylorate binding studies were already done in other plants, so we have performed in a sorghum also through wet lab experiment. Like it's a biochemical analysis we have done. But apart from that, we have done like for other benzamyl, TPA, MIA, only we have performed bioinformatic analysis only. Docking studies only we have done, but not in a wet lab.
No,
Uh,
no. I mean
that's
the
because you have not
energies.
performed
How?
any dynamic studies. You have not performed any. And the answer to your question, Renu, is we have not performed any dynamic studies in
No,
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 waste.
no,
dynamic studies we have not
no.
performed.
No, no. The binding energies, how different they are from each drug, ligand molecule you have checked. So that will help. I think. Is it okay, like to understand the question, or should I repeat that?
Yeah, that is okay, Renu. Like the binding sports and that is okay.
Have you tried to compare the energies? That's what I'm asking. Like how the binding is.
That we have not performed today. Like only we have identified the domains only, but binding scores and those all things we have not performed.
Okay, perfectly. But excellent with respect to other genomic studies and everything. It's really wonderful to know the phylogeneity and then and yeah, that's really good. I think Indrani is having some questions. Please.
Indrani, can you please ask question?
Hello, ma'am. Is my voice
Then
audible?
you have performed page for total
Yeah,
proteins
she asked.
isolated from plant cells.
Yeah,
Yeah, we have
she
performed
asked.
total proteins also in Indrani that I have not included in this slide. I have only given here only for co-immunopresentation, but we have performed total proteins also.
Okay. As in like from total cells,
Yeah.
total protein isolation from a plant cell. Hello.
Like
Hello.
from each part of the plant. Yes, like we have from root and leaf we have taken only two tissues we have taken.
Okay. Ma'am, may I know if you don't have any confidential interest, like what was the concentration of the proteins that you loaded from test tube stage?
Hello,
Hello. Hello.
hello, Indrani.
Indrani, maybe there is a network problem on her side.
Yeah,
Okay, she asked what was the concentration you loaded.
the protein.
Yes, ma'am. What was the concentration of protein you have loaded? Yeah.
2
We have
micrograms.
taken 5 micrograms.
For protein, we had taken initially 5, and for co-IP, we had taken 2 micrograms.
Yeah, five we have taken.
Okay.
My
Well, Hima ma'am,
question, can,
yes, yes, sir,
uh,
yes, sir. Please, please.
Sakshi, please call me Anil. Yeah, only doing
Okay,
inquiry.
okay, Anil.
My
Okay,
question
Anil,
to
please questions.
Dr. Hima, where can
Yeah,
we see plant genetics in down the lane of 10 years?
plant genetics in coming 10 years.
Yeah.
That's a bit difficult because for the plant people so far the like we are not getting any jobs first of all. To be frank, I say like plant genetics. I don't know because with the different we can combine like with the different groups together like with the omics, genomics, proteomics. With the coordination only, I think we can move further. In that way, plant genetics may have like, but alone, I'm not sure whether plant genetics it will go ahead.
Can we expect transgenics to be approved in India?
Can we expect?
Transgenic crops, food crops in the form of food crops, can be approved in India?
No,
Okay, thank you.
because it's a very lengthy and time-taking process to uh, take out the crop into the market. It's like with the transgenic approach, it's very difficult to get a crop into the market. You can get other alternative approaches of breeding.
What are the like we need to pay some crops in the according to the market.
Then your voice is breaking.
What are the hassles we need to face if you have any idea on this uh,
What is the
hassles we need to face in order to make the transgenic to the market?
marker-free approach? Like we have to grow the plants without a selection-free markers. We have to selection-free markers. Hello, Renu?
Yeah, actually, I'm asking that what are the like market is low or do we apply transgenic approaches to some plants? Then will there be any market for this or how it is actually? Do they have side effects or beneficiary effects? Any beneficiary, any nutritional value or anything?
As of
With
now,
a
in India,
selection
only
marker.
transgenic cotton is allowed, the transgenic one, especially the BT cotton, and
Yeah,
no other. We have developed BT brinjal, BT mustard, several things, and every day we keep on seeing the same thing in newspapers. There is a big
because
debate
with the
going on.
selection market,
Our
it's,
people
it's
has
having.
one minute more, please. Our
Yeah.
people has much apprehensions about transgenic crops, whereas other countries like in China, USA, Canada, they're having transgenic crops. In order to feed the ever-growing population, approval of transgenic crops in India would also become a mandate thing. As of now, we are able to manage, and the moratorium is up to 2023. The government, as the BJP government is there, they may extend the moratorium for another 5 years or 10 years. So it's a very difficult time for transgenic crops to be released in India as of now.
Because it would be having impact on the health also with the selection markers. So to first of all release that one, we need to have a selection free. Right?
Yeah. Can I talk?
Yeah, please.
Yeah. That was a wonderful presentation, Hima. And I don't need to say it again one more time. Part of PhD, part of postdoc. So part of PhD, I think we travel together. But
Yeah, yeah.
you are in
You
plan. I'm in
encourage me to
human.
PhD work.
So I'm the Anil is always there in your lab. But I always used to witness the kind of work that what you used to do during your PhD times. I have like a common question to both Anil and Hima because you worked in the same lab and you're talking about transgenics. Yes, there is always a lacuna in Indian point of view. I hope in the days to come the transgenics will come into picture and get established. But one point, like when Anil and Hima were doing in the lab, they used to do something based upon some implanter based approaches, right?
Yeah.
Dr. Anil and Dr. Hima.
Yes.
And is that particular implanted technologies is established in India or it is still in the mm, uh, taking a back seat? How do you address this technology? Yeah, please.
Yeah, thank you for the question. The thing here is, implanter is a, it's already established in adopsis, like in the form of floral dip and injection methods. And for the first time in the laboratory, we standardized that one for fruit injection. Straight away, we have taken the gene construct in the form of agrobacterium We had injected that one into the tomato fruit. We had incubated for different time periods like 24 hours, 48 hours, 72 hours, and at for 48 hours we got the transgenics. The percentage is very low, close to 1%. The transgenics percentage is very low. I mean transformation percentage is very low. But within 30 or 35 days, we are able to get even one transgenic or two transgenic, whatever it may be. On the other hand, the routine in vitro methods, they take close to starting from seed germination to getting a transgenic and I mean to confirmation of the putative transgenic, it takes close to 22 months, at least 18 months. 18 months. So we can save much of the time. And these days, as transgenics are not getting approved by the government, so people, not much people are involved or interested in working on the transition. Anyway, whatever they work, it will not be recognized by the government. So the companies, they have developed different, almost all transgenic food crops. Even though as there is a moratorium, they are not able to release them into the market. But the problem in Indian current scenario is we have a porous border from Pakistan as well as Bangladesh. Bangladesh, they had allowed BT brinjal, BT mustard. Our people feel that we are not eating any transgenics, but sometimes we are eating them. You can see the transgenic BT brinjal, transgenic BT mustard, some parts of in most of the parts of the India. The thing our people always feel is okay, India has banned that one, we are not eating. Especially the problem with social people, they keep on blaming the transgenics. There is no need for transgenics. Transgenics are safe to eat, especially if you think human being itself has elements from all other living species in the world. Our genes, we can say, even genes are contaminated with lizards, genes are contaminated with spiders, genes are contaminated with plants. So human being itself is a highly evolved by taking all the genes during the course of evolution. And especially in India, as there is no awareness or not much awareness on these things, they think that they are alien crops, and once they start eating them, they may face diseases. If we do not contain the transgenic markers, if we do not contain the transgenic markers, they are very safe to eat. And all the time they feel that they contain transgenic markers. DBT, Department of Biotechnology, especially with IBAC, it will check for that one. And if that RCGM, regional one for genetic manipulation, review committee on genetic manipulation, they approve only those crops which do not contain the selection marker. If the crops contain selection marker like kanamycin, it is little bit poisonous, but if it contains hygromycin, it is very toxic to your intestine cells. So you feel getting stomachache and all those things. So there is no need to worry because the government will check before releasing those things into the market. And there should be much awareness on these things because with ever increasing population, declining natural resources, this is the need of the hour to introduce transgenics into the market so that it will bring the another green revolution to feed the ever growing population. Yeah, thank you.
That was a wonderful explanation. Thank you so much. Yeah, thank you, Hima, for a wonderful and exciting talk. Yeah, yeah, thank you so much.
Thank you. Thanks.
Thank you, Hima ma'am. I would like to request Renuka ma'am, please give vote of thanks to Hima ma'am, please. Renuka ma'am.
Audible? Am I audible?
Ma'am, please give vote of thanks to Hima ma'am.
Yeah.